Hi, my name is Casey Cordova, and my group decided to modify an organism that lived within the coral. This organism helps the corals maintain their color. The organism is known as zooxanthellae. We will be modifying the zooxanthellae to help the corals maintain their color even though that the acid levels within the ocean increase. To begin, this is what a healthy coral reef should look like, with many fish and many colors, as seen in many movies and on Google. The CO2 absorbed in the atmosphere plays a big role in the combustion of ions which impedes calcification. When examining healthy coral, one can observe that the coral and algae depend on each other to survive. If stressed, algae leaves the coral. However, when the coral is stressed, it will expel the algae within its body. A bleached coral is a coral that no longer has algae within its body. The algae is what was giving the coral its color. Since there are no longer algae within the body, the coral is no longer colorful. As you can see in this picture, the coral is completely white.
Hi, my name is Taranisha Mejia, and I will be presenting our goal of our experiment. Ocean acidification is the changes in chemistry of the ocean having to do with increased CO2 and fossil fuels dumped into the ocean. This leads to slow growth of calcium carbonate. Corals secrete calcium carbonate to make a hard outer shell, also called tests. We are looking to find a way to make corals withstand the increased levels of acidity so they continue to be the homes and protection of many other organisms in the ocean.
My name is Rachel Lemp, and I'm going to be talking about the organism that's the genetic source for our gene. Knowing that acidophiles are able to live in a highly acidic environment, we hope to find a gene that allows for this pH regulation and place that into our zooxanthellae so that it can better regulate its pH in an increasingly acidic ocean. So this became our genetic source became the Deanaliella acidophila, which is a eukaryote acidophile. And this gene is basically a primary transporter for proton pumping. And the gene works by pumping the hydrogen ions to the plasma membrane. And this allows for the pH regulation because less of the hydrogen ions are kept within the cell and are pumped outside. And the more efficient that the organism is able to pump the hydrogen ions out of the cell, it'll be better adapting for that acidic environment, which is what we're hoping for. So this gene, this will be amplified from the Deanaliella acidophila genome by PCR and then cloned into our plasmid.
Hi, my name is Catherine Staple, and I'll be going over the methods part of our project. Using Deanaliella acidophila, we will grow the desired DNA. We will then isolate the DNA by using the selected primers that correspond to the DNA Deanaliella acidophila. The primers used are listed, primer 1 being 5', TT, GGTA, GTC, AA, GG, CCAT, CGC to 3'. The second primer being 5', starting with A, CA, TC, TCA, CTGA, T, GGG, TTTTC to 3'. We will then use PCR to amplify our DNA sequence. We will then take the gene sequence into our plasmid PHLUK for the Zooxanthellae D14. We will then take the plasmid with the sequence into a vector. With the vector inserted to our desired Zooxanthellae, we will use electroporation to bind the plasmid PHLUK to the Sambodium D14 DNA.
Hi, my name is Shelly Ariano, and I'm going to be presenting the plasmid map.
The plasmid that we are using is PHLUK. I found this plasmid on AdGene. It is 12,794 base pairs long. The main reason that this plasmid was chosen was that this plasmid is from the organism Symbiodin D14, which means that the promoters of this plasmid will work well once inside the organism. The plasmid also has an empty backbone, which is where we will put the DNA from Deanaliella. It also has the ampicillin-resistant gene, which will be useful for testing out if our plasmid is successfully inserted into our organism.
Hi,
I'm Danielle Ferdman. I'm going to be presenting the solving issue portion, which is on ocean acidification.
By modifying the zooxanthellae and adding a hydrogen pump, they will be able to increase their threshold of acidity, thus being able to endure the stress placed upon it with our increasingly acidic oceans, allowing to continue their process of producing calcium carbonate for the corals. With these corals back to full health, they can now again become the home of many organisms and restore their high biodiversity. If these corals are destroyed, many of these organisms that are at the bottom of the food chain will die, thus affecting the rest of the organisms throughout the entire ocean. This is beneficial to us medically because many of these organisms are being researched for cures to cancer, arthritis, viruses, and other diseases. From a business perspective, these beautiful corals and the fish that they bring produce much tourist business in these third world countries. From the way that they protect us is that with large protection from storms and crashing waves to prevent erosion.
Hi, my name is Adriana Rodriguez, and I will be talking about the ethical and legal implications that come with our long-term impacts that can be potentially irreversible. We won't know exactly how the corals will react, and if they are negatively impacted, it can damage our natural barriers that protect the shoreline from waves and storms and can damage shelters for numerous organisms. We won't know if one or multiple organisms can be affected, but we will try our best to prevent this from happening by conducting multiple thorough controlled experiments that can help prevent any disruptions to the natural environment. In terms of legal implications, we must gain a significant amount of permissions and funding from numerous organizations. Because ocean acidification is a worldwide problem, a fair amount of expenses are to be expected. We must gain permissions and grants from organizations such as the U.S. Environmental Protection Agency, National Oceanic